Hi, Ms. Patterson here. Today we're going to finish up our macromolecule unit over nucleic acids. So nucleic acid is going to be a form of hereditary information. It's going to allow you to store information, genes, for example, transmit information, help to express genetic traits. The two types are going to be RNA and DNA. Those are going to be my polymers of a nucleic acid. The monomer is a nucleotide. So my monomer is a nucleotide. My polymer will be DNA and RNA. This is a nucleotide. I can tell it's a nucleotide by three parts. I've got a phosphate group, pentose sugar, and nitrogenous base. So this is my polymer of nucleic acid. So this one's DNA. I can tell because it's double-stranded. It's in a helix. My three parts of a nucleotide. I have my phosphate group. Remember, our phosphate group is a functional group that we had to memorize. I have a pentose sugar. Pentose means 5. So I have 5 carbons. 1, 2, 3, 4, 5 carbons that are on this pentose sugar. I have a nitrogenous base. And we'll talk more about nitrogenous base here in a second. This pentose sugar, there's two types. Remember, I have DNA and RNA. So DNA is deoxyribose. And then I have ribose nucleic acid. So I have deoxyribose and ribose. I can tell which one I have from this pentose sugar. So see these numbers? I have a 1', 2', 3', 4', 5'. That's telling me which carbons are there. So it's a pentose 5 carbon. That's showing me where my carbons are located on this pentose sugar. The second carbon is the one we need to look at. The second carbon determines whether it's a deoxyribose or a ribose sugar. If I have a OH, I'm going to be a ribose, so it's ribose nucleic acid. If I'm missing the O, then I am DNA deoxy. Deoxy means there is no oxygen. So deoxyribose is DNA. So here I have ribose. I have my hydroxyl group, my OH group. Here I have deoxyribose. All I have is my H. So I show that in my formula, C5H10O5. This is showing that oxygen is there. Here I'm showing the oxygen is gone, C5H10O4. The next part of my nucleotide, so we've discussed phosphates, my pentose sugar, is my nitrogenous base. What you notice is there's a bunch of nitrogens here, and all these nitrogens are going to make my nitrogenous base. I also know it's a base. So what is going to make this a base? It's going to be this amino group that I have up here at the top. Remember that amino group pulls hydrogen protons out of a solution so it can make a solution more basic. So this is going to have a basic property to it. Complementary base pairing, I'm going to be able to pair certain nucleotides together. So this is going to have a nitrogenous base. This is going to be a specific nitrogenous base. But these are going to be attached to that amino acid. So G's are going to bond with C's, C's will bond with G's, A's to U's if I'm talking RNA, and then T to A with RNA. So this is base pairing. DNA will have these, RNA will have those. When I look at these, I can see here I have G to C. See these little dots? Do you remember what those little dots mean? Those mean that I have a hydrogen bond. So those hydrogen bonds are going to be there. So between guanine and cytosine, which is G's and C's, I'm going to have 3 hydrogen bonds. 1, 2, 3. So there's 3 hydrogen bonds between guanine and cytosine. If I look at my next one, I have cytosine and guanine. This will also have 3 hydrogen bonds. It's going to be the same. I have adenine and uracil. So this is saying it's a DNA complementary strand. Okay, I would have 2 bonds there, so 2 hydrogen bonds are going to be there. And if I have T, thymine, and adenine, I will also have 2 hydrogen bonds. Okay, so I have 2 hydrogen bonds here, one here, one here. DNA nitrogenous bases, I'm going to have adenine, thymine, guanine, and cytosine. So my pyrimidines will be a cytosine and a thymine. My purines will be adenine, guanine. And these will come together. So adenine and thymine are going to bond. They're going to be a complementary pair. 2 hydrogen bonds are going to put them together. Guanine and cytosine are going to be a complementary pair. And then I'm going to have 3 hydrogen bonds come between that. And if you ever heard of Kargaff's rule, Kargaff's rule is talking about complementary pairs. That's what it's talking about. Now I see DNA, double helix. I have 2 strands, adenine, guanine, cytosine, thymine. So I know that this is DNA. Over here I have a single strand. So that's RNA, ribose nucleic acid. And I can also tell because I have uracil. So this is letting me know this is RNA, this over here is DNA. My nitrogenous bases are going to come off the center. I'm going to have a phospho sugar backbone, a ribose backbone, the pentose sugar backbone that I'm going to have here, and my nitrogenous bases will be coming off the center. So uracil, RNA, thymine, DNA. All the other ones will be in both. So what makes a pyrimidine and a purine? So a pyrimidine is going to be 1 ring. When I have a purine is going to be 2 rings. So I have 1 ring and then I have 2 rings. The way I kind of remember, and this is how I tell my students to remember which is a pyrimidine and which is a purine, is you cut a pie, py, one at a time. I don't cut multiple pies in one go. I don't do a little cut and then cut a different pie. I cut one pie, I do it one at a time. So 1 pyrimidine is actually 1 ring. And the ones that are going to be those are going to be the cut letters. So cytosine, uracil, and thymine, these are all pyrimidines, which means they all have 1 ring. So thymine, uracil, cytosine are all going to have 1 ring. The other ones, adenine and guanine, will have 2. Another one I've heard before is a pyramid, which is 1 pyramid can cut you. If that helps you out, that's fine. But I've liked the pie one. I'm cut a pie one at a time, so I know CUT which ones those are. Some people will use pyramid can cut you. 1 pyramid can cut you. So same thing, cytosine, uracil, thymine, and it's doing 1 ring. How do you link a nucleotide to another nucleotide? Condensation reaction. Do you remember what the other name for that is? That's dehydration synthesis. So that means, remember, water is pulled out. So I have a hydroxyl group. I have another hydroxyl group here. This hydroxyl is going to get pulled off. One of these hydrogens will get pulled off, and water is going to leave. And when it leaves, it's going to bond to this 3rd carbon. And then I'm going to have what's called a phosphodiester bond. So this nucleic acid bonds to the one above it with a phosphodiester bond. So dehydration synthesis is what's going to make that possible. And that's how all the macromolecules are going to be put together. They're all dehydration synthesis, or a condensation reaction is going to happen. So again, this 3rd carbon is the one I want to look at. That one I'm going to bring in my phosphate group. This is going to have a hydrogen on there. That hydrogen is going to pull with this hydroxyl group, and then water is going to get pulled out. Then I'm going to have a phosphodiester bond for those. Okay. DNA has certain properties. It's going to be double helixed. I'm going to have anti-parallel, which means I'm going to have a 3' side, 5'. The other side is going to be opposite. So this 5 to 3, 3 to 5 is what we call anti-parallel. It's going to go one direction, the other one's going to be upside down, moving in the opposite direction. These are my paired groups, A's and T's, G's and C's. And this is also my, what they call a carbon, or a phospholipid or DNA backbone is kind of what they call this. I have phosphates, I have my pentose sugar, and these are going to produce those phosphodiester bonds. This is going to produce a covalent bond with my nitrogenous base, and then nitrogenous base to nitrogenous base is going to have a hydrogen bond going between those two spots. So this will unzip and break apart real easy, because remember, with a hydrogen bond, nothing's really being passed back and forth. They're kind of like magnets, and then this will unzip, and then it can zip back up. So DNA is able to pull apart and come back together real quick because of those hydrogen bonds. So here I have my pentose sugar and my phosphate. These are going to create this backbone. And then I'm going to have my complementary nitrogenous base. And if I asked right now, all right, this one has 2 hydrogen bonds. So what could these bases possibly be? Remember, the 2 hydrogen bonds are going to be between A's and T's. I know this is T because it's DNA. I'm double-stranded. So one of these would have to be thymine, the other one would have to be adenine. This one here, I have 3, so this would have to be cytosine and guanine. One of those would be cytosine and guanine because of that. So I can look here real quick. I can't tell which side's what, but I can tell you which ones would be here, which pairs I would have. So the sugar phosphate backbone is this part right here. And then this part here, my pentose sugar with my nitrogenous base is going to bond with the nitrogenous base on my anti-parallel strand on the other side, my complementary pair. So DNA double helix, 2 complementary strands. You have a main strand. This is going to be the one that is going to stay original. And then when I make a new strand, I can have a complementary strand that can come in. This is going to be one that's going to complement this other strand that's across from it. Again, they're anti-parallel. One's 5', one is 3', and I can tell which one is my 5'. My 5' is always going to be a phosphate end. So right here I have a phosphate, down here I have a phosphate. That is my 5' end. Down here is 3. Okay, this is my hydroxyl end, and then this is going to be my hydroxyl end up here. And this is my 3'. And the reason why it's 3 and 5 is this 3 end is attached to that 3rd carbon. This one here, this phosphate, is going to attach to that 5th carbon. So this is why it's 5' and 3', because it's attaching to those specific numbered carbons that are here. So 1, 2, 3, 4, 5, those carbons are numbered. So this is my 3 end because I'm attaching to that 3rd carbon. This is my 5th end because I'm attaching to that 5th carbon. So that's why it's 3, 5'. RNA, single strand, again, uracil. This is what I'm going to use. This will leave the nucleus. This is going to go out. This is going to help code for proteins. So when we talk about proteins, they're coded from DNA. So this is going to go, it's going to find a ribosome, and it's going to make proteins based on the code of the RNA, and this will be a complementary strand from the DNA. So I have my nitrogenous bases, my sugar phosphate backbone is going to be right here. Okay, that's going to be nucleic acids. That's it for all the macromolecules. If you guys have any questions, let me know. Talk to you later.